We're going to go ahead and move on to our keynote address. And I would like to introduce Dr. Dale Corbett. It's my absolute pleasure. Dr. Dale Corbett is Professor Emeritus of the Neurosciences in the Department of Cellular and Molecular Medicine at the University of Ottawa, and affiliate investigator of the Neuroscience Program of the Ottawa Hospital Research Institute. Dr. Corbett's laboratory is internationally recognized for pioneering research on the protective effects of prolonged mild hypothermia that culminated in the use of therapeutic hypothermia in the treatment of cardiac arrest and perinatal asphyxia. Dr. Corbett has made pivotal translational discoveries regarding the importance of implementing early and more intensive rehabilitation following stroke that have been incorporated into best practice guidelines for stroke rehabilitation. His work has been cited over 18,000 times. Dr. Corbett uses novel forms of rehabilitation, exercise, and drug treatments to enhance neuroplasticity and stroke recovery. He is part of an international preclinical clinical collaboration seeking to determine how much rehabilitation should be given and when should it be given to optimize post-stroke recovery. Related work focuses on the potential of biomarkers for predicting effective doses of rehabilitation to enhance recovery. Other ongoing research focuses on neuroprotective interventions to minimize or delay vascular cognitive impairment, and a funded trial is ongoing in that regard. I know that I have certainly followed Dr. Corbett's work closely over the years, as it has provided a roadmap of important concepts and intellectual ideas as we conceptualize interventions to accelerate post-stroke recovery for every patient. I'm certainly excited to have you here with us, Dr. Corbett, to share your thoughts and look forward to your keynote address.
Okay, thank you very much, Mona, for the very nice introduction, and thank you for inviting me to participate in this very exciting conference. Just share my screen here. There we go. So I'm going to be talking to you today about early stroke recovery and where are we headed. But before I really get into where we're headed, I think it's important to know where we are right now. So in the last several decades, there have been major advances in neuroscience and our understanding of brain plasticity. But in spite of all these advances in new knowledge, there's been fairly limited translation of research to improve stroke recovery. So our field is kind of like at a crossroads right now, seeking direction. And I think this is because we failed, at least in part, to answer some very fundamental questions. So for example, when should therapy be given? What therapy or therapies should be given? How much should be given? And who should receive this therapy? And therapists really don't know the answers to these questions. So what I'm going to do today, or try to do, is in my talk about our very basic research using animal models of stroke that address all these questions, particularly in the early time period after stroke. So let's look at some of the changes that happen in the brain over time once a stroke has occurred. If we look, this is a kind of complicated slide, but if we look at these different time periods going out to the chronic period, which is 6 months after stroke, we can see that in the hyperacute period between 0 and 24 hours, it's characterized mainly by cell death and inflammation. A few days later, this starts to give way to what we call endogenous plasticity processes, a variety of adaptive changes to the brain injury, which we think are critical for improving impairment and function. So these time periods have to be kept in mind when we do interventions, to be thinking of what's actually going on during each of these time periods. So it seemed quite reasonable that after a stroke, what you would really like to do would be to start encouraging recovery as soon as possible. And this was really the basis of quite a famous trial, the AVERT trial, a very large trial conducted by Julie Bernhard and colleagues out of Australia. So AVERT stands for a very early rehabilitation trial. And in this trial, patients were given very early and frequent mobilization, actually less than 24 hours after their stroke. But unfortunately, this led to less good outcome, not better outcome, but less good outcome at 3 months compared to usual care. So the question here is, why did AVERT fail? Well, this slide, I think, shows some of the reasons why it did fail. And I want you to pay close attention to this slide because there'll be a pop quiz on it at the end of my presentation today. So when you have a period of ischemia or stroke, it induces a whole host of pathological mechanisms which can worsen cell death. It creates an energy deficit, inflammation, there's an excitotoxic response, and the brain tries to counteract these pathological processes in order to protect itself. So let's just take the example of excitotoxicity. In order to counterbalance this excitotoxic action, which could kill more brain cells, one of the things that happens is that the inhibitory neurotransmitter GABA is increased, its levels increase in the brain in order to counteract or oppose this excitotoxic reaction. So let's just see what the consequences are of manipulating GABA levels in the brain very early after stroke or a little bit later on. So this work comes from work done in Tom Carmichael's lab. In the first few days after stroke, inhibition increases in the peri-infarct region, that region right around the stroke. So this inhibition increases in order to protect the brain. If you reverse this GABA inhibition by giving a drug less than 3 days after stroke, it worsens the injury and leads to poorer recovery in the animal models. However, if you give the same drug after 3 days, it improves outcomes because long-term, you really don't want these high levels of GABA inhibition because it can interfere with recovery. So when you look at the animal studies that have been done over the years, they consistently show that very early rehabilitation or induction of exercise or other interventions results in increased injury or poorer behavioral outcomes. So you have to be very, very careful in the first couple of days in that hyperacute and early acute phase after stroke of what is done because it's possible to worsen injury. So given this, when should we actually start rehabilitation? So let's take a look at some of the molecular changes that occur in the brain after stroke. So when a stroke occurs, we see that after a few days, there's an increase in a number of proteins and genes that are associated with neuroplasticity. So these are things like brain-derived neurotrophic factor, GAP43, which among other things encourages sprouting of new connections. So these are things that have been shown to improve recovery. But notice this is rather a time-limited change. It rises up quickly over the first few days, hits a plateau, and then after about 30 days, and this is in the animal models, it begins to decline. And notice how this curve compares very well to the spontaneous motor recovery in rat models of stroke. So initially the behavior is very suppressed, the motor behavior, and then over a period of time, it begins to spontaneously recover, matching quite nicely this kind of profile. I also want you to notice that there are also growth inhibiting factors in the brain. Initially after the stroke, they're expressed at very, very low levels. But over time, they begin to increase as well. so that after about 30 days post-stroke, the growth inhibitory factors become to predominate over the growth promoting factors. So this suggests that there's this period of heightened plasticity in here between 5 to 30 days when it may be optimal to induce or to use rehabilitation. Okay, so that's fine. What kind of rehabilitation can we use to best take advantage of this period of heightened plasticity? Well, one of the very best things to increase plasticity in the brain of experimental animals, and I would argue also humans, is something called environmental enrichment. And this is a phenomenon whereby animals are housed in these stimulating complex environments where there are opportunities for them to engage in different sensory motor activities, socialization, and cognitive stimulation by introducing new objects, and so on. The environmental enrichment creates a permissive regenerative state by enhancing multiple neuroplasticity processes, which we can see here. So you can see environmental enrichment will increase cortical thickness, cause an increase in dendritic spines, more synapses, axonal sprouting. It will increase the levels of growth promoting factors. So across many different types of brain injury, environmental enrichment has been shown to improve gross motor function and cognition. It's important, though, to realize that environmental enrichment is not very effective in promoting recovery of skilled reaching, a common deficit in human stroke. This requires some type of task-specific training. So, what are the active ingredients of environmental enrichment? This slide shows quite nicely what the enriched environment looks like. And you can see that multiple animals are housed together, giving the opportunity for socialization. They're very social animals, just as are humans. So this provides them with that opportunity. Also, by varying the different types of toys and various apparatus in the enrichment cage, it challenges the animal's sensory motor system. So in this case, the animal is standing on a very unstable platform. So it really challenges the animal's balance. And after stroke, the animals can engage in these different activities as they become able to challenge their sensory motor systems. We produce cognitive stimulation by putting in new objects at different times. The animals are very curious and they'll investigate these new objects. We can put them in different places, put them on different floors. And one of the key things about the enriched environment is that it provides the animals with novelty and really kind of a play atmosphere. Though I'm anthropomorphizing here, it's fun for the animals to be in these stimulating, enriched environments. So a number of years ago, we developed a paradigm which we've called enriched rehabilitation, which consisted of housing animals in environmental enrichment. And then for several hours a day, they get task-specific reach training, where they're put in a different apparatus and they're able to grasp these very yummy little sugar pellets. But the only way that they can get them is to use their impaired limb. So they go into the chamber and we set the level so it's fairly easy for them to grasp the pellets, and then gradually over time, we make it progressively more and more challenging by lowering the level. And the rat is actually quite an excellent model of upper limb recovery, which is the main interest in my laboratory. And these photographs show quite nicely the dexterity of these animals. If you look at this rat, it's grasping this small 45 milligram sugar pellet, making a pincer grasp like this. So they're very skilled. And when you produce a stroke, like kind of strokes that people do, you get the same kind of impairments in reaching and grasping as you see in humans. This slide shows the vasculature on the top of the brain, the bottom of the brain of a rat, and the distribution of the blood supply is very, very similar to what you see in people. And we produce our strokes experimentally, and we do this by injecting a compound, endothelin-1, which is a vasoconstrictive agent. So we can inject this anywhere in the brain to produce strokes in the cortex, in subcortical structures, or both the cortex and subcortical structures. And we can vary the size. We can produce small strokes, medium, or very large strokes. And so we can do any kind of stroke that you see in humans. As in human stroke studies, we use a battery of behavioral tests to assess functional recovery. But today I'm going to be just talking to you about the tests that we use the most and that we like the best because it's highly sensitive. And this is the staircase reaching test. In this test, animals are trained to reach for food pellets in this specialized staircase chamber. They go into the chamber and crawl up on top of this platform. And on each side, on the left and on the right, are these series of 7 steps, each containing 3 food pellets. The only way that they can get the pellets on the left is to use the left paw. The only way they can get them on the right side is to use the right paw. So you kind of get the picture of what's going on here. We produce a stroke on the right side, and they are markedly impaired at reaching for the pellets on the left side. So we can quantify this very precisely. So we train the animals for a couple of weeks prior to inducing a stroke, get a very stable pattern of behavior. Then we do the stroke and look at how their post-stroke behavior compares. And then we can look at that over time and assess functional recovery. So in this study, which is one that we did quite a few years ago, we trained the animals in the staircase task, and then we induced a large middle cerebral artery stroke. We assessed the animals a few days after the stroke to gauge their impairments and balance the groups. And then we started our enriched rehabilitation paradigm, which consisted of environmental enrichment and daily reach training. The control condition was just standard social housing of the animals. And so we started the enriched rehabilitation 14 days after the stroke. And we were curious to see if this combination therapy would increase recovery, because these are very large strokes. We assessed the animals 4 weeks after the start of rehabilitation, and again 9 weeks later. This shows the results of that study, and on the y-axis you see the percent reaching success in the staircase. So the sham non-stroked animals, are performing, at very, very high levels, 100%, or even a little bit higher than what they were at baseline. The 2 stroked groups, the ones that got enriched rehabilitation or the ones that got standard housing, both are markedly impaired, going down to about 20% of baseline levels after the stroke. However, the group that gets the enriched rehabilitation shows highly significant recovery, improving by 30 to 35%, even with these very large strokes, where the animals that were just exposed to standard housing conditions show virtually no improvement. We then went on to do subsequent studies where we varied the time at which we started the enriched rehabilitation after stroke. And what we found is that there is a critical or sensitive period for rehabilitation when this intervention is most effective. When we started the enriched rehabilitation, any time between 5 to 14 days, it worked very well, produced significant recovery of upper limb reaching in these animals. But when we delayed it by 30 days, it was really not nearly as effective. It produced very limited benefits. So this suggests this window of time if you start the enriched rehabilitation or you do exercise interventions within this time window, it's a very good time to capitalize on all these different plasticity processes that I talked about earlier, and we see improved recovery. If you delay by 30 days, then it doesn't really work very well. We think because these growth promoting factors are beginning to decline and the growth inhibitory factors are starting to predominate. So this is the critical period for post-stroke recovery in these animal models. And this has been replicated, many, many times. So that sounds kind of interesting and exciting. And so you say, well, that's well and good, but is there a sensitive or critical period in human stroke recovery? Does it matter when you start rehabilitation in humans? So turns out that a phase 2 clinical trial testing an optimal time for motor recovery was conducted in humans by Alexander Dromerk and colleagues. And this is the critical period after stroke study, or CPASS study. And, this is, quite something, because here's a clinical trial, the very first one that I know of, that was modeled after an animal or preclinical study. It was modeled directly after our publication from 2004. And what Dromerk and colleagues did was to have their patients, have extra task-specific therapy starting in the acute phase of after stroke, less than 30 days, at 2 to 3 months, the subacute phase, or they delayed greater than 6 months. So very similar, albeit a slightly different time scale as in our animal studies. What they found was that the most robust upper limb improvement was found in the subacute, followed by the acute group, and there was no significant improvement in the chronic group whatsoever. So these findings confirm the results in the animal studies, showing a time-limited period of post-stroke heightened neuroplasticity when rehabilitation is most effective. This doesn't say that rehabilitation cannot be beneficial at longer post-stroke periods, but it shows that there's a period of heightened plasticity. So this is pretty exciting that there's translation from an animal preclinical to the clinical stroke recovery trial. So the environmental enrichment has shown to be quite efficacious in producing stroke recovery in the animal models, but you might be thinking, I don't really see how that could be implemented clinically. We don't have the same kind of control. So why don't you just do more reaching and forget the environmental enriching? Maybe just a greater dose of reach training would be beneficial. So we did those experiments where we dissected the different components of enriched rehabilitation. So we had 4 groups of animals in this study. And again, it was with the middle cerebral artery occlusion strokes, which are shown here, very large strokes. And we had sham animals who had no stroke. And so they show just the stable reaching performance. We're showing here is the absolute number of food pellets retrieved here. So this is pellets retrieved on the y-axis. And then we had the 3 stroked groups. One was housed in enrichment. One just got lots of extra reach training. And then the other group was the enriched rehabilitation group, which you'll recall is the combination of environmental enrichment plus task-specific reach training. So let's look at the performance of these different groups. So the group that only got enrichment showed very little recovery in the staircase test of skilled reaching, a little bit of improvement from where they were in the first week, but fairly minimal. The group that got lots of extra reach training did somewhat better, but still, not anything to get terribly excited about. The group that showed the strongest improvement was the Enriched Rehabilitation group. Much more dramatic and more robust recovery in this group. So clearly, there's something about the combination of environmental enrichment and reach training that conveys much more recovery than any of the individual components. So it's the combination that's really important. So given that the enriched rehabilitation combination is so important and so effective, how does it actually change the brain? What's it doing that these other individual components are not doing? Well, in our early studies, we looked at the brains of animals who were housed in this enriched rehabilitation paradigm versus standard social housing. And what we did was to stain the brains of these animals with a Golgi stain, which stains the motor neurons in the cerebral cortex. So you can really see the fine architecture and structure of these motor neurons. So this is a motor neuron in which the animal had a stroke and was exposed to enriched rehabilitation. This is an animal that had a similar stroke, but had this type of standard housing. And I think you can see right away, there's a huge difference in these motor neurons. There's a tremendous increase or proliferation of these dendrites in the group that got enriched rehabilitation compared to animals that had the same kind of stroke, but were housed in this environment. And as it turns out, the animals that showed the best or the most robust proliferation of these dendritic arbors showed the best recovery. So there's a strong correlation there. We then went on to dissect the components further and take a look at other parts of the brain in a different way. So instead of doing a Golgi stain, we used a stain for a marker called FosB. And what this does is it's a marker of use-dependent neuronal activity. So it can tell you what parts of the brain are being activated by an intervention. So it's kind of like functional imaging. So what we did was, again, a similar kind of experiment where, we had the animals exposed to very large strokes. And then what we did was we instituted these different interventions after the stroke, beginning about 7 days or so after the stroke occurred. So animals either had the standard social housing, they were put in enriched environments, or a group was given daily reach training, or we had the enriched rehabilitation group. So 4 different groups. And then we sacrificed animals from each of the groups at different intervals in the early stroke recovery period to see how the brain was changing. So the question was, what did each of these interventions do to the FosB staining? And it was very interesting what we found. Here on the right-hand side of this slide, you see the strokes, and I'm showing you the pattern of FosB activity on the non-damaged side, just because it's a little easier to understand. So the enriched rehabilitation increased the density of FosB staining throughout the cortex, but notice this banding of heavy staining right here. This corresponds to layers 2 and 3 in the cerebral cortex. This pattern was very different than what we saw with just an environmental enrichment or reach training alone. They produced less dense staining in the cortex, and we didn't see this banding pattern in layers 2 and 3. We've since gone on to look at other experiments and look at other parts of the brain to see how enriched rehabilitation is changing the brain. So this is a cross section of the rat brain in the midbrain showing the red nucleus here. And this is a blow up of the different types of cells within the red nucleus. And this shows the pattern of FosB staining. And what we see is that the enriched rehabilitation, shown in red, increased the FosB staining in this rostromedial magnocellularis part of the red nucleus, which gives rise to the rubrospinal tract. So the point of this slide is that the enriched rehabilitation is inducing widespread changes in activity in both cortical and subcortical brain structures, which we think are giving rise to the improved recovery that we see post-stroke. So how does all this work with enriched environments compare with what's being done clinically? Well, we know in the first few weeks after stroke that patients are alone about 60% of the time. And during their waking hours, they're inactive about 75% of the time. They're resting in bed or sitting in a chair. The lack of stimulation and exercise and lack of socialization compared to environmental enrichment is quite striking. And when you ask patients their experience of what it's like to be in rehabilitation, these are the kinds of reports that you get from them. They report being bored and inactive. They've been spent a lot of time just waiting for something to happen. And many of them express capability and desire of doing more. So clearly we need to change the post-stroke environment, making it more like what we have in the animal studies. So in the early post-stroke phase, how does dose affect recovery? That's another important question that we need to answer. Well, the AVERT trial set out to determine if more rehabilitation would be better. So this was a trial led by Carolee Winstein. And what happened in this trial was patients were given an extra 30 hours of upper limb rehabilitation over a 10-week period. Unfortunately, this didn't provide any added benefit. So the authors concluded that more is not necessarily better. So why did AVERT fail to provide, benefit? Why wasn't more therapy better? Well, if we look at most human therapy sessions, the number of upper limb repetitions is quite low. In humans, it's roughly about 30 or 32 per therapy session. Contrast that with what we do in our animal studies, where the paradigm we use now, we give them several sessions per day, each about 15 minutes long, with break periods between each session. And doing it this way, we're able to get the animals making skilled, purposeful, reaching movements up to 400, 500 times per day. And this leads to significant recovery of function post-stroke. So I think the answer for the reason that AVERT and other trials like it have failed to show benefit is that the dose of rehabilitation is not sufficient to induce all these different neuroplastic mechanisms which are essential for functional recovery. Whereas in the animal studies, we exceed this threshold and we can actually measure these changes in the brain and see that the doses of rehabilitation that we're giving are actually changing things that are associated with recovery. So given the failure of the AVERT trial, a number of researchers, including John Krakauer, Kathy Stenier, Bibelo, and Corbett, have argued that the recovery in humans is mainly determined by spontaneous biological processes and compensation. So sure, people get better, but it's due to spontaneous recovery and compensation. And these recovery patterns seem to follow a 70% proportionality rule, which really means the function regained is about 70% of what was lost, or measured early after stroke. So if you look at, do a Fugl-Meyer early after the stroke and you assess the patients, you'll see that based on that, you can predict where they will be, and it'll be about 70% of where they were, prior to stroke. This recovery occurs irrespective of the type or amount of rehabilitation given. So this is kind of what it looks like, the proportionality rule. So this regression line here, and so this is the Fugl-Meyer upper limb measure of deficits in humans. And you can see that based on the early Fugl-Meyer score, you can predict the level of upper limb recovery that they eventually achieve on the Fugl-Meyer. And most of the patients fit this line quite well, the 70% proportional recovery rule. There are, however, some patients that don't fit this line or fit this rule, and these are called non-fitters, whereas the ones that do show 70% proportional recovery are referred to as fitters. The non-fitters tend to be patients who had larger and more severe injury. So we wondered, if we're using our animal models to really talk about recovery, we really need to know if we see similar kinds of things as shown in the human studies, because if we don't show the same kind of fundamental process, then maybe our animal modeling really isn't going to be that useful for predicting what might happen in humans. So we did an assessment of nearly 600 animals that had been done in various studies in my laboratory over many years, and assessed them for their early post-stroke impairments to see if we could predict their recovery using the same kind of equations that were used in the human studies. And indeed, we also saw the 70% proportionality rule apply. And we saw the fitters who fit the rule or the regression line, and we also saw non-fitters. And we saw many more non-fitters because in the animal studies, we never exclude animals. So animals that have very, very severe impairments that might get excluded in human studies typically get included in animal studies. So we saw more non-fitters. So it looks like the 70% proportionality rule is common across humans and animal models of stroke. So then we did studies to look and see how such things as size and location of the stroke would affect recovery. And here we're measuring changes in pellet retrieval. And we see that with purely cortical strokes, that there was a greater propensity for recovery versus strokes that affected both cortex and striatum. Importantly, we also saw that task-specific rehabilitation made a difference to post-stroke recovery in animals. So unlike the eye care study, task-specific rehabilitation had an impact on recovery, and it would allow us to predict how much these animals would recover. So this was all retrospective analysis. So we took this information and then developed an algorithm based on the initial impairment, these biomarkers of initial impairment and infarct volume. And from this, we could prescribe different doses of rehabilitation to give each animal based on the severity of their initial impairment and the infarct volume. And when we did this, what we saw was a nice dose response improvement in recovery. And the animals who met their prescribed dose of rehabilitation, that is, they did what they were supposed to do, all showed significant recovery. If the animals didn't meet their prescribed dose of rehabilitation, for whatever reason, they didn't show recovery. And interestingly, The animals that benefited most from the rehabilitation were the animals with the moderate to severe injury, where the 70% proportionality fitter animals, it didn't really matter to them whether they got rehabilitation or not. They sort of all reached the same level. So for the message from this work is that for the most severely affected subjects, the ones that really need the high dose rehabilitation are the ones that should be getting more rehabilitation. So the greater the impairments, the more rehabilitation they need. So similar type of modeling and development of algorithms for predicting recovery potential have been done in humans too. And so this is some of the work that's been done by Kathy Stenier and colleagues in New Zealand. And she's developed what are called PrEP algorithms, and PrEP stands for the predict recovery potential for patients. So what they do in their clinic is they go through a series of steps which involve clinical, neurophysiological, and imaging biomarkers. So the SAFE score is based on shoulder abduction and finger extension. So if they score higher than 8 on this scale, they will tend to see complete recovery in patients like that. If these patients show less than 8 on the SAFE score, then they deliver transcranial magnetic stimulation, determine if their motor evoked potential is present. If there are, then they're predicted to make significant recovery. It won't be complete, but it'll be significant. However, if the motor evoked potentials are absent, then they will then do imaging to determine the asymmetry in the cortical spinal tract fibers. And what's been shown there, if we look on the right-hand side of this slide, is that if you have severe cortical spinal tract loss, as shown here by these dark diamonds, then you will not see very good recovery in the upper extremity Fugl-Meyer score at 3 months. If more of the cortical spinal tract is intact, then you'll see a better eventual recovery on the Fugl-Meyer later on. So with the imaging, you can get an asymmetry index in terms of cortical spinal tract integrity. So if it's very symmetrical, what that means is that the damage side is so sparse, the number of fibers remaining, that there will be very limited recovery. And for these patients, it's probably best to encourage compensatory movements rather than task-specific therapy. So what's coming from the animal work and the human work with these algorithms is that one size of rehabilitation does not fit all. What's missing from the human work is that we still don't have the dose response for rehabilitation that we have in the animal models. We know how much in the animal models is needed to produce recovery. We don't know that yet in humans. When we can finally determine what that dose is, then it'll be possible to develop more powerful algorithms that are even more personalized and I think more effective for producing recovery in patients. So what does the future hold? Well, I think based on what we've seen in the animal models, that the rehabilitation in the future will be given early, but not too early. We'll not go into that early 2 to 3 day period. It will be optimized for dose. We have to get away from 32 repetitions per session and get it more in line with the animal repetition or dosing, which is possible by increased frequency. And we also have to be concerned about the content, what's actually being done in the rehabilitation session. Is it actually task-specific therapy, or is there a lot of wasted time where the patients are really not doing anything? And that's a problem with just using time as a measure of rehabilitation dose. Rehabilitation will need to be delivered in more stimulating, enriched environments. And work underway on this is being done in humans, mostly in Australia. Heidi Janssen and others are doing this kind of work with encouraging results so far. Rehabilitation will be personalized using biomarkers to match each patient's unique functional and injury profiles. So stroke location, stroke size, a number of other amount of cortical spinal tract remaining, and so on. However, even if we do all these things, rehabilitation will not be sufficient to produce game-changing gains in recovery. It'll help, but to get the kind of TPA-like moment in stroke recovery, we're going to need adjunctive interventions. And this is for the future, more creative and innovative interventions, recovery promoting drugs. This has been underdeveloped. More selective forms of electrical stimulation, brain-machine interfaces will be needed to augment and direct plasticity enhancing effects of rehabilitation. In closing, I would just like to thank all the wonderful staff and trainees in my lab over the years who contributed to all the studies that I talked about today. A number of my collaborators at the University of Ottawa and the University of Montreal, and the funding agencies in Canada that provided research support. So thank you very much for your attention, and look forward to answering any questions you might have.
Wonderful, Dr. Corbett. Thank you so much.